Hello everyone. In this video we're going to understand the regulation of gene expression in bacteria like E. coli by taking three different examples of operon systems, the lactose, tryptophan and arabinose. So let's start from the basics. What is an operon? The operon consists of a set of structural genes which form one transcriptional unit which is under the control of a single promoter and other regulatory sequences. Like example, the binding site for a regulatory protein. So all the combination of these forms a functional unit which is referred to as an operon. That is, all the structural genes of an operon can be switched on or off in a coordinated fashion, and a polycistronic mRNA is transcribed. So let's take them one by one. So first we are going to talk about the lactose operon or the lac operon, which can be defined as a set of coordinately regulated genes in E. coli which are involved in the utilization of lactose from the growth medium. Lac operon is an inducible system. That means that this set of genes are coordinately switched on in the presence of lactose, but this induction is however dependent on the presence of glucose. The operon would not be switched on in the presence of glucose even if lactose is added. This forms the basic regulatory working of the lac operon. The operon consists of three structural genes: LAC-Z encoding the enzyme beta-galactosidase, LAC-Y beta-galactosidase permease, and LAC-A beta-galactoside acetyltransferase. The structural genes are under the control of a promoter P-LAC, which contains the binding site for RNA polymerase as well as for the regulatory proteins, the repressor called as LAC repressor and the activator CAMP, which is nothing but catabolite activator protein complex. The binding site for the repressor is the operator, while that for the activator is called as the cap binding site. The repressor protein is encoded by the regulatory gene LAC-I, which is not a part of the lac operon. It happens to lie adjacent to the lac operon, as you can see in this slide. Now we are going to go through the mechanism of lac operon regulation by understanding the situation under four different media conditions. First, when lactose is present and glucose is absent, state of lac operon is on. In the absence of lactose, the lac operon is maintained in a repressed state, where the repressor binds to the operator and prevents the transcription of operon. As lactose is added to the medium, it is brought into the cell by permease, which is coded by LAC-Y, and converted to allolactose by beta-galactosidase enzyme, which is coded by LAC-Z. The allolactose binds to the repressor, which in turn prevents it to bind to operator. As now operator is free, RNA polymerase transcribes the polycistronic RNA and lac operon is induced. The second state, when both lactose and glucose are absent, state of lac operon is off. As mentioned earlier, in the absence of lactose, lac operon is maintained in a repressed state. As there is no lactose, the repressor is free to bind to operator and repress the transcription of lac operon. Now we are going to understand the effect of glucose through these other two conditions. 1. When lactose is present and glucose is also present, state of lac operon is off. In this condition, the lac operon is present in the medium, but because of the presence of glucose, the cell is not able to take up this lactose inside. This is because glucose inhibits the uptake of lactose via permease through cellular mechanisms. So as the lactose is not taken up, the repressor is free to bind the operon and hence lac operon is not induced. The second state, where lactose is absent but glucose is present, state of lac operon is off. Again in this situation, lactose is not present in the media at the first place itself, and the uptake is also being repressed by glucose. Hence the lac operon remains in an off state. The tryptophan operon, also known as TRP operon, is involved in responding to the cell's need to produce the amino acid tryptophan. Now, the operon consists of a set of 5 structural genes, as you can see in this slide, TRPE through TRPA, which encode for the 3 key enzymes needed for the synthesis of tryptophan from chorysmic acid. The regulatory region of the TRP operon consists of a major promoter P1, which drives the transcription of the 5 structural genes as a polycistronic RNA. Overlapping the P1 promoter is the operator region O, to which binds the repressor protein. And in addition to the promoters and operator, there is a third control region called TRPL. The TRPL, which is the first region to be transcribed under P1 promoter and encodes a 162 nucleotide long mRNA leader sequence. There are 2 major regulatory mechanisms in TRP operon. First, the repressor binding, and second, the attenuation. We will discuss both of them in detail in the upcoming slides. But the logic behind it is the regulatory circuit operating at the TRP operon is designed such that if the cells have enough tryptophan, the operon is switched off. This is logical, as the cell already has enough of tryptophan. It would not waste its energy in synthesizing the enzymes needed for tryptophan biosynthesis. As the tryptophan is used up by the cell and the internal level falls below a threshold, the operon is switched on to reinitiate the process of tryptophan synthesis. Therefore, in the presence of low concentration of tryptophan, the operon is on, while a high concentration of the same switches it off. The operon is thus a repressible system. One of the control points of the TRP operon is the repressor protein, which is encoded by the TRPR. The product of the TRPR gene is an aporepressor, which functions as a repressor only when bound by tryptophan, which is called as a corepressor. Now, this functional repressor moiety binds to the operator to downregulate the operon. In the absence of enough tryptophan in the cell, the repressor is non-functional and thus cannot repress the operon. Like the LAC repressor, the TRP repressor also regulates via negative control mechanism, but is activated in the presence of the signal, that is the tryptophan. Now, in addition to this negative regulatory mechanism, TRP operon also employs another control mechanism called attenuation. Now, attenuation is a mechanism of transcriptional control that involves premature transcription termination. Now, in this slide, we are going to explain the structure of TRPL and how it forms the basis of attenuation. When the TRPL region is transcribed, the RNA formed has the potential to base pair in short stretches due to the presence of inverted repeats as shown in the figure. Region 1 can base pair with region 2, region 2 can base pair with region 3, and region 3 can base pair with region 4. This region of the transcript can thus take up 2 different secondary structures. Under the conditions when region 1 pairs with 2, region 3 pairs up with 4, as 2 is not available to pair with 3, the structure formed by the pairing of the regions 3 and 4 is that of a row-independent termination. This terminator blocks further transcription. The terminator structure is formed as long as region 2 is not available for base pairing with region 3. Under conditions when the region 1 is prevented from base pairing with region 2 pairs with region 3. The row-independent terminator is not formed, allowing transcription to proceed beyond into the structural genes of the TRP operon. Now, in the next slide, we will understand how the levels of tryptophan dictates the event of attenuation and regulation of TRP operon. The level of tryptophan in the cell dictates whether the terminator would be formed or not. This is controlled by the presence of an open reading frame in the TRPL region, which encodes for a 14 amino acid long peptide. The translatable region overlaps within the region 1 of the TrpL transcript as given in the figure. Now, the important feature of this ORF is the presence of two codons encoding the amino acid tryptophan. Following transcription initiation from the TRP promoter, the TrpL region is transcribed and subsequently translation of the ORF is initiated. By the time the RNA polymerase reaches region 2 of the TrpL, the ribosome is in a position to translate the two TRP codons. The time taken to incorporate the two TRP amino acids decides whether attenuation would take place or not. Now, there can be two possibilities. Under conditions when the tryptophan levels are low in the cell, the ribosome is stalled in the region 1 as it has to wait for a longer time to incorporate a tryptophan molecule. While the ribosome stalls, the RNA polymerase transcribes the rest of the TrpL region. As the region 3 is transcribed, it immediately base pairs with region 2, which has already been transcribed. Now, thus, region 3 is not available for pairing with region 4, which prevents the formation of the termination signal. The RNA polymerase thus moves ahead and transcribes the structural genes of the Tryptophan Operon. Now, when under conditions of high levels of tryptophan in the cell, the ribosome does not stall in the region 1, but moves ahead encompassing the proximal portion of region 2. While RNA polymerase continues transcribing regions 3 and 4, region 3 cannot base pair with region 2 as the ribosome makes this region. Now, region 3 thus pairs with region 4, leading to the formation of row-independent terminator signal. This results in the termination of transcription of what is called attenuation before the structural genes TrpE through TrpA can be transcribed. The attenuation system is thus able to sense the intracellular concentration of tryptophan and modulate the regulation accordingly. Now we are going to talk about the Arabinose Operon. E. coli cells can use arabinose as the carbon and energy source. The arabinose is brought into the cell with the help of a transport system involving several gene products. The proteins involved in this uptake are encoded by genes which are not part of the Arabinose operon. The genes involved are ARAF, G, H and ARAE, as you can see in this slide. While ARAF, G, H constitute an independent operon under the control of a promoter PFGH, ARAE is an independent gene with its own promoter PE. The enzymes needed for the catabolism of arabinose are encoded by ARA-B, A and D. These constitute the ARA operon with the common promoter PBAD that transcribe the polycistronic message. This transcription is regulated by the ARAC protein, which is encoded by this ARAC gene lying upstream to the ARA promoter. The RAC protein has a dual function. In the presence of arabinose, it acts as a positive regulator to stimulate transcription from the PBAD promoter, while in its absence, it helps in negatively regulating this promoter. It also functions as a negative regulator for its own gene. Now, the RAC protein does this by binding to two different sites upstream to these promoters, that is I, which is made up of two half repeats I1 and I2, O1, that is O1L and O1R, and O2. Now, the RAC protein has two binding domains, one a DNA binding pocket and the another arabinose binding pocket. These two domains are connected by a flexible linker region. Now, the RAC dimer can take up different conformations depending on whether arabinose is bound to it or not. That is, in the absence of arabinose, the linker region is stretched out in such a manner that the two DNA binding domains of the dimer can reach out to two far off half sites, that is O2 and I1. This binding is further facilitated by the looping out of this, as you can see in this slide, this 210 base pair DNA region. However, when arabinose is bound to the ARAC protein, the outreach ability is not that flexible, Under these conditions, the DNA binding domains can bind only to the closely spaced direct half repeats, that is I1 and I2. This binding releases the DNA loop which was formed earlier, and the negative control is converted to a positive regulation as the ARAC protein now interacts with the RNA polymerase and helps it in transcribing the genes of the ARA operon. Now, the binding of the ARAC protein, it positively regulates the ARA operon, but when the high levels of ARAC protein is attained in the cell, it binds to the half sites O1L and O1R, which leads to the turning off of the ARAC gene. Hence, it negatively regulates its own promoter PC. So, in summarizing it, the Arabinose Operon uses a positive regulator, that is the ARAC protein, to turn on the operon in the presence of arabinose. But in its absence, there is an appropriate interaction of the positive regulator is inhibited and the operon is turned off. Now it is also further repressed by the formation of a DNA loop. So this slide contains the references from which I have taken pictures that were put into this video. These deal with operon systems in more detail. If you want to read them in more detail, want to know about the experiments, how they find out what was the mechanism behind each operon system, you can go and very well read them. Fourth one is a paper which is easily available online. That's it. Thank you for hearing me out, and if you want more such videos, stay subscribed to this channel.